By doing a peppermint diffusion demo. So diffusion is the movement of ions, or just basically particles, from an area of high concentration to an area of lower concentration. So in this case, the stuff is the color and the dye in the peppermint. So we're going to be placing the peppermint in the Petri plate, so then it can diffuse with the water.
In this lab, we're going to be demonstrating how diffusion works in a cell through dialysis tubing, which represents the plasma membrane of a cell. The cell membrane allows certain things to diffuse in and out of it, including based on cell size and charge. But in this particular lab, we're just doing cell size.
So we have a beaker of water here, and we're going to be adding a glucose and starch solution to the inside of the bag. And we are adding IKI, which is a color indicator. It turns black and blue in the presence of starch, and we're adding that to the water solution.
Now we're using a glucose test strip to see if there's any glucose in here, and there is, because it is a glucose starch solution.
And so
as you can see, there's glucose indicated. We tied off the bag so it's a closed system, and now we can start the experiment. What do you guys think will happen?
So while we're waiting for the reaction to occur, what we're expecting to see is the glucose is a small enough molecule, it's a monosaccharide, that it should be able to diffuse outside from the inside of the bag to the water. And the IKI, which is also a very small molecule, should be able to diffuse into the cell from the water. The starch, however, is a polysaccharide, which means it's a bigger molecule, and it won't, and because it's so big, it will be stuck inside the bag and will not be diffusing into the water. And so we are going to be using glucose test strips and the IKI indicator to see whether or not this actually happens.
Some of the IKI diffused into the dialysis tubing, turning the starch inside black. And the starch did not diffuse outside of the tubing because the molecules are too big to leave.
In the first lab with the peppermint, we focused on how diffusion works as a whole throughout a cell. In the second experiment, we specifically focused on diffusion within a cell, and in this lab with the banana, we're going to focus on extraction of DNA. DNA is found inside the nucleus, and to extract that DNA, we need to break the plasma membrane in order to reach it. We use a banana in this experiment because a banana is a polyploidy, which means it has a lot of extra DNA, so it's easier to extract. So the first step in this experiment is to put the banana in a plastic bag, and we're going to mash it until there are no lumps left.
No lumps! Ooh, very mashy.
We can speed this up if we have to.
I think this should be good, right?
Yeah. Okay. So as you can see, there are no lumps, and yeah,
and it looks like this. So the next step is to mix soap and salt, which we already did, into a beaker. And what the soap does is it breaks down the plasma membrane, and the salt loosens up the DNA, so it'll be easier to extract.
So the next step is to add the soapy water mixture in with the mashed banana.
It's very appetizing. Now Olivia's just massaging the bag, getting everything together there.
So for our next step, we're going to pour the filtrate back into the beaker with a filter to make sure no lumps get through. We have to stop every couple of seconds to make sure the mixture is actually getting through.
So if you can see,
Okay, next we're going to take ethyl alcohol, which is this, and pour it into the filtrate that we poured back into the mixture. When we do this, we should see the DNA and the alcohol separate because the DNA weighs less than the alcohol and should float. And we're going to let this sit for a minute to give it time to separate. As you can see, the DNA is floating to the top and the alcohol is sinking to the bottom.
here's a little better view.
And there's a clear distinction between the DNA and the alcohol.
Thanks for watching our demonstration of diffusion and extraction of DNA.
As you can see, biology is like no other science. You get to do the coolest experiments and it's so much fun. We hope you guys enjoy bio next year. Bye!